Good day, this is Professor Will, CBMD PhD. Today's Monday, November 1st, 2021. It is 7:34 and 48 seconds, p.m. Eastern Standard Time. Today's research, bone defects. So let's hop to it here. Non-thermal atmospheric plasma stimulates new bone formation in critical bone defects of animal model. Date October 29, 2021. Source, Osaka City University. A recent project has revealed new bone formation and critical bone defects of an animal model with an exposure of pencil-type non-thermal atmospheric pressure plasma surgery. Researchers found the usefulness of NTAPP in healing of bone fractures using a pencil-type NTAPP in animal bone defect models. NTAPP is considered a new therapeutic method, as it has been shown to accelerate cell growth when applied at low enough levels. In an ambient atmosphere, it can generate highly reactive oxygen and nitrogen species, RONS, which can be directly exposed to biological targets like cells and tissues. Indirect treatments have shown the potential advantages of plasma in supporting the creation of stem cells that cause reactive oxygen species and inducing osteogenic differentiation and bone formation. However, as the team points out, there is no report on directly using NTAPP for bone fracture therapy. The research group developed a pencil-type plasma device that can effectively generate and deliver RONS to an animal model with a well-established critical bone defect, allowing the team to search for the optimal irradiation conditions. Comparing groups that were irradiated with NTAPP for 5, 10, and 15 minutes to control groups with no plasma administered, micro-CT images at 8 weeks showed the 10-minute treatment time as the most successful bone regeneration, with 1.51 times larger bone volume than the control group. However, micro-CT images cannot determine whether a bone defect has been filled with new bone, tissue, or both. Therefore, the team also ran a histological analysis and confirmed bone defects in the groups treated with plasma were filled with new bone and there was no inclusion, such as fiber tissue, and gap that was observed in the control group. And like other forms of therapy, the biological effect of plasma depends on the treatment dose delivered into the targets. Although future research will be needed to clarify why the study saw the most bone regeneration during the 10-minute treatment period, it is understood that surface wettability promotes greater cell spreading and adhesion to biomaterials and implants. Researchers wondered if something similar was occurring, where they saw a strong generation of new bone, and they found that compared to the control group, bone surface of the plasma-treated group, and statistically and significantly more hydrophilic. And while there is still much to be explored and understood in terms of treatment dose, for the first time ever, the direct application of room temperature and atmospheric plasma to a living body has seen a positive result. The research team hopes the plasma device they developed can become something used during surgery to bring the bone regeneration effect of NTAPP to various medical fields. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.